Whole genome sequencing, also known as full genome sequencing, complete genome sequencing, or entire genome sequencing, is the process of determining the entirety, or nearly the entirety, of the DNA sequence of an organism's genome at a single time. This entails sequencing all of an organism's chromosomal DNA as well as DNA contained in the mitochondria and, for plants, in the chloroplast. Whole genome sequencing has largely been used as a research tool, but was being introduced to clinics in 2014. In the future of personalized medicine, whole genome sequence data may be an important tool to guide therapeutic intervention. The tool of gene sequencing at SNP level is also used to pinpoint functional variants from association studies and improve the knowledge available to researchers interested in evolutionary biology, and hence may lay the foundation for predicting disease susceptibility and drug response. Whole genome sequencing should not be confused with DNA profiling, which only determines the likelihood that genetic material came from a particular individual or group, and does not contain additional information on genetic relationships, origin or susceptibility to specific diseases. In addition, whole genome sequencing should not be confused with methods that sequence specific subsets of the genome. Such methods include whole exome sequencing or SNP genotyping. As of 2017, there were no complete genomes for any mammals, including humans. Between 4% to 9% of the human genome, mostly satellite DNA, had not been sequenced. The DNA sequencing methods used in the 1970s and 1980s were manual. For example, Maxam-Gilbert sequencing and Sanger sequencing. Several whole bacteriophage and animal viral genomes were sequenced by these techniques. But the shift to more rapid, automated sequencing methods in the 1990s facilitated the sequencing of the larger bacterial and eukaryotic genomes. The first organism to have its entire genome sequenced was Haemophilus influenza in 1995. After it, the genomes of other bacteria and some archaea were first sequenced, largely due to their small genome size. H. Influenza has a genome of 1,830,140 base pairs of DNA. In contrast, eukaryotes, both unicellular and multicellular such as amoeba dubia and humans than A. dubia, however, their genome size far outweighs the genome size of individual bacteria. The first bacterial and archaeal genomes, including that of H. influenza, were sequenced by shotgun sequencing. In 1996, the first eukaryotic genome. In 1999, the entire DNA sequence of human chromosome 22, the shortest human autosome, was published. By the year 2000, the second animal and second invertebrate genome was sequenced, that of the fruit fly Drosophila melanogaster, a popular choice of model organism in experimental research. The first plant genome, that of the model organism Arabidopsis thaliana, was also fully sequenced. By 2000, by 2001, a draft of the entire human genome sequence was published. The genome of the laboratory mouse Mus musculus was completed in 2002. In 2004, the Human Genome Project published an incomplete version of the human genome. In 2008, a group from Leiden, the Netherlands, reported the sequencing of the first female human genome. Currently, thousands of genomes have been wholly or partially sequenced. Almost any biological sample containing a full copy of the DNA, even a very small amount of DNA or ancient DNA, can provide the genetic material necessary for full genome sequencing. Such samples may include saliva, epithelial cells, bone marrow, hair, seeds, plant leaves, or anything else that has DNA-containing cells. The genome sequence of a single cell selected from a mixed population of cells can be determined using techniques of single-cell genome sequencing. This has important advantages in environmental microbiology in cases where a single cell of a particular microorganism species can be isolated from a mixed population by microscopy on the basis of its morphological or other distinguishing characteristics. In such cases, the normally necessary steps of isolation and growth of the organism in culture may be omitted, thus allowing the sequencing of a much greater spectrum of organism genomes. Single-cell genome sequencing is being tested as a method of preimplantation genetic diagnosis, wherein a cell from the embryo created by in vitro fertilization is taken and analyzed before embryo transfer into the uterus. After implantation, cell-free fetal DNA can be taken by simple venipuncture from the mother and used for whole genome sequencing of the fetus. Sequencing of nearly an entire human genome was first accomplished in 2000 partly through the use of shotgun sequencing technology, while full genome shotgun sequencing for small genomes was already in use in 1979. Broader application benefited from pairwise end sequencing, known colloquially as double-barrel shotgun sequencing, as sequencing projects began to take on longer and more complex